Good day, everyone. The emergence of witches' broom disease in Trinidad and Tobago over the last 7 years is posing a serious threat to local cocoa production. This year, the disease prevalence and severity island-wide was particularly high, with a number of farms reporting 100% pod losses during the 2020 to 2021 pod season. Up until now, witches' broom disease has been a distant second to that of black pod disease, which continues to ravage the cocoa industry. So, the fungus Monilioptera perniciosa attacks meristematic, vegetative, and reproductive tissues of cocoa, which leads to the distinctive, distorted, misshapen shoots and flower cushions, which we call brooms, as well as infecting pods, which can also be misshapen, show early ripening, and develop blackened lesions, ultimately destroying the beans inside. The International Cocoa Genebank Trinidad has an intertwined history with witches' broom disease. The impetus for prospecting of wild cocoa in Latin America by F.J. Pound in the late 1930s was the search for resistance to this deadly disease, which at that time resulted in heavy crop losses island-wide. The pathology team has been actively involved in laboratory screening and evaluation of the germplasm collection for resistance to witches' broom disease. And this current disease epiphytotic presented an excellent opportunity to correlate findings of the field with that obtained in the lab. The ultimate objective is to find reliable sources of resistance to witches' broom disease, which can be used in breeding to obtain genotypes with resistance. The specific objectives were to determine the incidence ascertained as the percentage of trees infected per accession, to determine symptom severity by calculating the average number of vegetative brooms per accession, average number of cushion brooms per accession, and average percentage of pods infected with the disease. In addition, we wanted to determine the concordance of susceptibility of accessions to vegetative and cushion brooms, and to determine whether the sources of resistance to witches' broom disease identified in the field correlates with those identified in greenhouse studies using the agar droplet method. How did we go about doing this? Phenotypic evaluations for witches' broom disease on 500 accessions were conducted between March to May 2021, immediately following the crop season. The selected accessions were distributed across 5 fields in the gene bank, and these trees within each of the 500 accession plots were pruned and fertilized during July to September of the previous 2020 growing season. This allowed the emerging new flushes to become uniformly infected. Each plot consisted of 4 to 16 clonally propagated trees of each of the 500 accessions. Accessions were selected based on the diversity of ancestry as outlined by my colleague David Gopalchan in a previous study. The aim being to capture accessions as wide and as diverse a span across all genetic groups as possible. Figure 1 illustrates the diversity of these 500 accessions and their ancestry being calculated using admixture software using a 2.7 million NIPS panel. Each individual tree per plot per accession was assessed for total number of pods per tree, number of disease pods across all accessions, number of vegetative and cushion blooms, including cherry moyas, per tree across all accessions. Other diseases present on the trees were also recorded. And the uniformity of infection across the 5 fields was first assessed by selecting 10 highly susceptible accessions from each of the 5 fields and determining the percentage trees infected per accession. The results, based on percentage infected trees, all 5 fields developed 100% infection. This gave a clear indication that there was homogeneous or uniform spread of infection across all fields at the gene bank. While this is utterly bad for the collection, it was good for the screening for witches broom resistance. This uniform spread allowed us to clearly separate out the goods from the bads, so to speak, and negated concerns about uneven pathogen distribution from field to field. Disease incidence in the field varied significantly. By analyzing the proportion of replicate trees per accession which exhibited symptoms, we got a clear indication of the prevalence of disease among the clones. Of the 500 accessions, 10% or 51% were recorded as showing no disease symptoms based on shoots, flower cushions, and pods screened. These have been designated in this study as putatively resistant. 172 or 57% showed 100% infections, where every replicate tree per plot per accession developed symptoms. These accessions were classified as highly susceptible to infection establishment. However, the number of vegetative and cushion brooms varied among these accessions, as can be seen from the histogram. Symptom severity analyses using Glimanova indicated that there were significant differences between individual accessions for total percentage brooms as well as percentage vegetative and cushion brooms. However, out of these, the 20 most resistant accessions, namely those accessions with few or no symptoms, were selected for further comparative studies. In addition, the epiphytotic incidence of witches broom disease at the gene bank presented an opportunity to select accessions which performed well under extremely high disease pressure. It also presented an opportunity to correlate the findings of field and laboratory screening. Preliminary correlations validates the results of the lab screening thus far. Additionally, 2 levels of resistance were identified from the field screening. We found the first level at the level of disease incidence, and the second level of resistance at the level of severity, where we found variations in broom numbers, which range from a few to more than 100 per tree, per accession, even in accessions with 100% of trees per plot. Additionally, accessions which were susceptible to cushion brooms were largely not the same as those with susceptibility to vegetative brooms. So some accessions with large numbers of vegetative brooms exhibited no cushion brooms and vice versa. This suggests that resistance to vegetative and cushion brooms may be governed by different mechanisms. Of course, further validations of this is required. And we also found some preliminary concordance between high levels of resistance scored in the field and those identified in lab screening. This study was also able to successfully identify 51 putative accessions with resistance, and these will be further validated by greenhouse screening. So that brings me to the end of my presentation. As always, I want to thank everyone for joining our CARDS webinar series, and a special thank you for the support from the Government of Trinidad and Tobago and the Cocoa Research Association of the UK for their continued support of the Cocoa Research Centre. In addition, I'd like to especially thank my colleagues Vindra, Kishan, and Leon for their support in doing this study under very difficult circumstances earlier this year. Thank you very much.